So I'm just going to say it again because I don't remember what I said. So here we go. So neuropeptides, and I'll show you another picture of a neuropeptide. Neuropeptides are the largest by size, not like the largest group, but they are the largest by size of the neurotransmitters that we've learned. And remember that they are between 5 and 40 amino acids stuck together with peptide bonds. And the receptors that they interact with are generally, oh yeah, I remember I was going to tell you guys about something. Delta kappa mu receptors, that's just what they ended up calling the receptors. So listen to this because it kind of gets interesting. There is one that we know of and a couple more that we've been, mu1 we know is related to dependence. So can you see how pharmacology might want to find something that interacts with like pain perception and doesn't activate the mu1 receptor? So part of the reason I'm trying to get you guys used to the idea of thinking about receptors is because a lot of clinical practice is based on this kind of like pharmacology says you need to know what receptor you want to interact with and what receptor you don't want to interact with. So I'm not going to do at all a comprehensive job of teaching you about receptors, but I want you to at least have heard of them before. So the mu1 receptor we know is related to dependence. I think there's also one other one that they think is now. So, and here's the other issue that we run into. Remember that whole dependence process that you start with dopamine, right? The dopamine reinforcement pathway that starts with the nucleus accumbens and the VTA and then goes forward to the prefrontal cortex, right? That sort of pathway that you can reinforce. And of course, we know that like methamphetamine and cocaine, those all directly activate the dopamine pathway, but this is something that we need to talk about. Opioids reinforce themselves by causing release of dopamine also. So it's not in the opioid pathway, but it causes a parallel release of dopamine in the reinforcement pathway. And I want us to think about that evolutionarily before we go any further, right? And then we'll look at treatments for dependence and addiction and opioids. So you can probably imagine why if you saved your baby from a saber-toothed tiger, you would want to reinforce that behavior, right? Does that make sense? So what happens is as you are, for instance, diminishing pain perception, right? And making you feel like you have great capacity to do this and reducing fear, right? You are also saying that, yes, and you're a badass, and the next time that happens, you should do it again. So you can imagine with those two things that drugs that interact with opioid receptors tend to be relatively pleasant for a lot of us because evolutionarily it's a reinforcement pathway. And of course, that relatively pleasant can directly correlate to the predisposition for addiction. So we're going to talk about that. So when opioids are released, that dopamine is also released in the nucleus accumbens and can go forward. So I think I mentioned this to you guys last time. Did we talk about morphine and heroin and all that kind of stuff? So there's a whole bunch of opioids. They are mostly, yeah, go ahead.
Is PCP considered one of those?
No, it is not. It activates the reinforcement pathway, but not because it's an opioid.
Okay. But
yep.
Like, would it have? Because like I know endorphins, like they
give you this like
reduce the pain perception.
Yeah.
But then you have like PCP that like you're like on a.
It's just a different chemical mechanism in the brain that has a similar outcome. Okay. But it's not the same neurotransmitter system. Okay, so I want to talk briefly about this because you guys will find, I find this interesting. I'm probably not going to test you on it. So I am going to test you on this part. But so opioids tend to be one of our most effective analgesics, right? Pain reducers. But of course, they're also highly habit forming. And I just want you guys all to know that the habit formation that occurs with opioid use doesn't have to occur with opioid abuse. Okay, so you could, for instance, be prescribed opioids for whatever, and you are using them exactly as prescribed, and you can activate your reinforcement pathway, and you can actually become chemically addicted to the opioids with normal usage. So that's important for you guys to understand. So opioid addicts are not people who are using it inappropriately necessarily, and they're certainly not people who are using it illegally necessarily. And you probably know, and I probably mentioned to you guys last time, that most opioid addiction occurs starting with a legal prescribed dose. And then what happens is because of downregulation, either the legal prescribed dose no longer does what you need it to do, right? And you can't get more, and you still have massive amounts of pain, right? And it's also reinforcing itself with the dopamine reinforcement pathway. Does that make sense to you guys? Okay, so this is important for us to understand. So I want to show you guys. I thought you guys might be interested because I looked this up. One of my students asked me about it a couple semesters ago. I'm not going to test you over this, but it is interesting as hell. Okay, so I put this table down here, not because I'm going to ask you everything off the table, but because the same sets of questions kept coming up. So I thought I would gather it for you in a place for you guys to look at it. So one of the things that students often ask me about was this. So the whole opioid addiction overdose treatments, you've got this on the bottom of yours, right? Okay, so I'm not going to ask you these, but I thought you might want to know how these kind of things work. So when somebody has opioid addiction, several different treatments exist. One of them is methadone. Have you guys heard of methadone treatment? Methadone is federally regulated. You usually have to go through a methadone clinic. And what methadone really does is replaces an opioid that's short-acting and will cause drug-seeking behavior, which tends to be disturbing in your life, right? With a long-acting opioid that acts for 24 hours and produces an analgesic effect. And you are still an opioid addict, but you are a functional opioid addict with methadone. Does that make sense to you? And sometimes that's really the only choice for people. Like opioid addictions are very, very difficult for a lot of people to get over, but methadone actually gives you a way to have a long-acting analgesic effect. It's not particularly pleasurable. It's not as fun as heroin, for instance, but it basically keeps the cravings at bay and the drug-seeking behavior at bay. And it's legal, right? It's federally regulated and legal. Then some other ones that have come out within the last 10 years that you guys may find interesting. Have you guys heard of Suboxone? If you have anybody in your family or friends who is an opioid addict who's trying not to be an opioid addict, sometimes they will choose this one. Suboxone is a 4-to-1 ratio. It's an interesting thing that it does. What it is, it's a partial opioid agonist, so it interacts with some opioid receptors, but it's a mu antagonist. Remember that mu1 receptor? It's an antagonist there. So what it's trying to do is to give you the analgesic effect and decrease the dependence effect. And in addition, it also has something in it that will make you completely gut sick if you do take opioids at the same time. Does that make sense? So it's sort of got this negative reinforcement impact. Does that make sense to you guys? So that's a maintenance medication for somebody who's trying to get off of opioids. Okay, and then the last one, this one is a rescue medication. Have you ever had any experience with this, Scott? Naloxone?
Oh, for sure.
Oh, okay.
Yeah.
Okay, go ahead.
Do they usually call it just Narcan? Like Narcan something?
Yes. Yeah. Okay, so naloxone. I don't have any personal experience with this one, but I do know how the physiology works. So naloxone is an opioid antagonist. And an opioid antagonist, and it's a really, really high affinity for opioid receptors. So if you're in the middle of an overdose or a potential overdose, and you take naloxone, it's a high affinity opioid antagonist. It blocks the receptors. It's used for emergencies. It is not a maintenance medication. It's a rescue medication. It doesn't have any pain relief, but it can keep you from, for instance, your respiratory system will shut down with too many opioids. Does that make sense? Go ahead.
Yeah, it's crazy. My friends are both nurses, my roommates, and they are like Narcan people like every night. Because a lot of times we don't know
Really?
why people are overdosing or like why they're like shutting down. So sometimes like we'll just Narcan them.
Yeah.
See if they're like overdosing.
This medication is available now over the counter from a pharmacist. If you have someone in your life who is an opioid addict, it is not unreasonable for you to figure out how to use it. You can figure out on the package. You can go on. There's videos about how to use it. It's a rescue medication. And how is it pretty effective?
Yeah. Like I said in the last class, if you can get there early enough. Like,
Yeah.
Because a lot of times that overdose, it knocks out the respiratory drive. If
Yeah.
you get there on time, then you're going to then you're going extensive treatment. But
Right.
other than that,
And interestingly, if they have a lot of opioids in their system, sometimes you're going to have to administer it more than once. Because as it breaks down, then they've still got a lot of opioids in your system. You might have to administer it again before their respiratory system. So we'll shut down. Go ahead.
So I actually work at a retail pharmacy.
Oh, yeah, you do. Yeah.
And then nowadays, and that with the law claims, they require the pharmacist to consult with patients
Awesome.
to get opioids, so that way they know it can be. opioid.
Really?
Yeah, now they require the pharmacist to do that before we sell it to them.
Before you sell someone like a new opioid prescription?
Mhm, yeah.
Oh,
So,
that's
yeah,
awesome.
they
That's really
require the
responsible.
pharmacist to consult with them about that and make sure like they know that Narcan does exist, so that way they can get just in case.
Yeah, okay. So, interesting little tidbit about this one. There are some areas of the country in which ambulances don't carry it and cops won't carry it because they think that this is a negative reinforcement, and basically, if you're going to overdose, it's not our job to save you.
That's insane.
Yeah, I know. I'm like, oh, okay. So you're gonna teach them a lesson. Well, they're dead, so you didn't really teach them anything that's going to. So anyway, that I thought you guys might. Oh, sorry. I thought you guys might find that interesting, just because people always ask me how those work. So I decided to go look it up and figure it out. Go ahead.
You know, there's something kind of like Suboxone that it's like a shot though, and it lasts for a month.
Oh, really?
Anything, it just makes you sick, and you don't get
And it's like Suboxone, but it's a long-acting one.
back. It lasts a month.
Fascinating. I wonder what that one is. I'll have to look that up. Okay, the last of the neuropeptides. I don't know a whole lot about any of these, but some of my students told me you might want to introduce these, so I'm going to go ahead and put them and tell you that they fit into neuropeptides. They're not opioids, okay? CCK is one that regulates gallbladder contraction, and that ends up being important for people with gallbladder problems. This one's interesting. This is orexin, which is a hypothalamus neurotransmitter that regulates your sleep-wake cycles, and this is one of the treatments for narcolepsy that is being studied. And then there is this one. This one is one that is not an opioid, but is a neuropeptide, and it's called substance P, and substance P is involved in a different portion of pain that we don't usually treat. So remember, we have pain sensation, transmission, and then perception. Opioids treat perception, right? We don't really have much that treats sensation, so substance P is being studied as another possibility of an analgesic that doesn't activate the dopamine pathway. It's not in use yet. Okay, endocannabinoids are next. So endocannabinoids are their own category, and I find these totally fascinating. And since less than a week away, you are going to be voting on whether medical marijuana should be legalized. We should have a conversation about this. And then I'm not trying to tell you what decision to make. I'm simply trying to tell you how to find reputable information and the basic physiology behind it so that you can make your own grown-ass adult decisions. Okay, so endocannabinoids, let's talk about the neurotransmitter system first, and then we'll talk about THC. Okay, so endocannabinoids, the only one that you need to know, it's not the only one that exists, is a neurotransmitter called anandamide. And anandamide comes from the Sanskrit ananda or ananda, I don't actually speak the language, does anybody else? Which means bliss. And here's the fascinating thing to me about this. This neurotransmitter was only discovered in 1992, which I know you guys are younger than me, but 1992 literally feels like yesterday to me. And how many hundreds of years have people been using cannabinoids, phytocannabinoids, plants? Hundreds and hundreds of years, and we didn't even know that this neurotransmitter existed. And then fascinatingly, once we figured out that the neurotransmitter existed, we figured out that there were receptors all over the brain for these. They're like ubiquitous receptors. So it's a lipid-soluble neurotransmitter, which is unusual, right? Most of the rest of them are water-soluble, which means that if you can get it into the bloodstream, it's going to get into the brain, right? Okay. So the receptors are called cannabinoid receptors, CB1 and CB2, and they are just everywhere in the brain. There's just so many of them. It's fascinating to think that we didn't even know those receptors were there when they're so ubiquitous in the brain. So it's a lipid-soluble neurotransmitter, and it is involved in pain signaling. So it's a different kind of analgesic. So we have pain sensation. Right now we don't treat that very much except for like local anesthetics, right? And then we have pain perception, which is what opioids do. And this is pain signaling, pain transmission, right? So what it does is interrupts the pain signaling. It's also involved in some other things, and that's why you will get side effects by using it, just like you do with everything else, for an analgesic. It's also involved in working memory. It's involved in eating and sleeping pattern, and it's involved in pleasure and food rewarding, but not because of the dopamine pathway. Importantly for you guys to understand, it does not tap into the dopamine pathway in any way we've ever found. So this whole food rewarding is a food rewarding that's not related to dopamine, okay? Also important, it does not depress your respiratory function, and so it is not life-threatening in the way that opioids are. So this is an important thing for you guys to understand. It doesn't have any impact on the respiratory centers of your brain, and people do not shut down breathing and die because of marijuana, okay? I'm not saying that nobody's ever died. Somebody probably fell off of something, right? But it doesn't shut down your respiratory system. So it is not immediately physiologically life-threatening in the way that opioids are, okay? In addition, people don't die of marijuana overdoses, okay? You might feel like you want to die, but you're not going to die of marijuana overdoses.
Did you hear about that sick throw sickness though that people get?
The throw-up sickness?
What? So there's a new disorder from people who smoke way too much weed, like outrageous amounts, and they'll just feel nauseous and like they're gonna throw up all the time. And I guess my friends have been getting in the ER a lot because they just can't stop like feeling like they're gonna throw up.
Interesting.
And
I wonder if it's a
not smoke
strain.
weed for a couple months or something, and then they can't. But these people are probably smoking like all day, like outrageous amounts. But it's weird. I guess.
I wonder what that's
a
about.
new thing I just started seeing.
I don't know what that one's about. Okay, so let's talk about THC, and let's talk about medical marijuana use, and then you guys make your own blasted decisions. But please freaking vote regardless, because how are the old people doing at running the country? Do you like what's going on? You do? You're just delighted? You're just delighted with how we're running it, right? You like all of our decisions. Okay, so CB1 and CB2. So here is anandamide, which is an endocannabinoid. Here is THC. What they have is enough molecular similarity and shape that they activate the same receptors. Okay? All right. So THC and anandamide. There's other compounds in there that are also neurologically active. What happens is THC is a very weak anandamide agonist. So it behaves kind of like anandamide, but it's lipid soluble, right? So it gets relatively quickly into the brain and spinal cord and can have an analgesic effect within a matter of minutes, okay? And you don't need vast quantities of it because, of course, there's so many receptors. So that's important for you guys to understand as well. And the analgesic effect is involved in pain transmission or pain instead of pain perception. So it's a weak anandamide agonist. It is one of the next wave of analgesics that people are considering using. So what's of course on the ballot next Tuesday here in Utah is not a legalization of recreational marijuana, but a legalization of medical marijuana. And in addition to being a possible treatment as an analgesic, it's also an anti-seizure medication that is really, really effective in a lot of people. So put that in your hat. Now, the delivery mechanism, smoking is not good for anyone, and we all know that, right? So the delivery mechanism is something that you have to consider when you're dealing with this. There are also other delivery mechanisms. Of course, once it's legalized, right, in states that have legalized it, you can have like little chewables and little gummy bears, and you can put it into food and all that kind of stuff, and you can circumvent the smoking as a delivery mechanism. There's also other ways to do it. In addition, and this is important to understand, when and if it is legalized at a federal level, you get more research dollars into it, and one of these two receptors has an analgesic effect and the other one has psychotropic effects, and you can breed strains that do one or the other. So, which is going on all over in Canada. Oh yeah, they're already doing it. It's just a little bit more difficult to get in the United States because of federal regulation. So importing across, you can't go across state lines, and you can't go across boundaries of countries either. I mean, not legally.
Do you understand like how CBD interacts differently than THC? Like why that is like
We can talk about it, but not right now. It's kind of complicated. But if you want to come talk to me about it, we can. Okay, so the THC in marijuana is a weak anandamide agonist. It's not the only weak anandamide agonist that we've ever found. Weak anandamide agonists are also found in cocoa and chocolate. So part of the reason that last night was so fun for you is because you got a little high. Right? And so it's not THC, guys. It's just another weak anandamide agonist. It's not THC as in cocoa and chocolate. And just to sort of round out the conversation as you guys go figure out what the hell you want in your own lives, I also want to tell you that when marijuana is no longer federally illegal, we can do a lot more research with it. And there are some potentials. One of them is that anandamide, not THC, as far as I know, THC hasn't been tested. And this is only in a test tube or in a petri dish. This is not in a person yet. But it's kind of interesting because they figured out that once they found anandamide, somebody decided to stick it on growing breast cancer cells. And anandamide actually inhibits breast cancer growth in vitro. So trying to figure out whether any anandamide derivative or agonist would be useful for treating breast cancer in vitro. And we're not there yet. So these are your endocannabinoids, your anandamide. So anybody have any questions? Okay. So go argue with your family and figure out how you're going to vote, and then definitely vote. Some way, shape, or form, because otherwise other people get to make decisions for you. And I don't trust other people, do you? No. Okay, so miscellaneous neurotransmitters. I'm not going to tell you anything about anything major about these, except that weird crap we discover is neurotransmitters. So carbon monoxide, what will happen to you if you breathe too much of that in? You're dead, okay? But your brain makes it and releases it as a CNS neurotransmitter. I don't know, okay? Nitric oxide, not nitrous, which is the giggly laughing gas at the dentist that everybody loves. But nitric, it makes you say, oh, that's so sad. Because it's like you can get high and nobody judges you. You're like, oh, that was great. Nitric oxide is different, and nitric oxide gas is also a neurotransmitter, and it gets lots of money because that's the one that's implicated in erectile dysfunction. And guess how much money people will spend to keep getting erections? All of it. Just all of it. How much money do you have? There, that's how much you get. Okay. So lots and lots of research goes on into that one. Okay, so the next thing that we're going to do is kind of irritating and boring, but we need to do it. So the next thing that we are going to do is to talk about receptor functions and receptors in general. We usually do this I used to do it with endocrine system, but endocrine is complex enough, so I put it over here. So what I want to do is I want to talk briefly about the categories of receptors that exist, because again, once you get into nursing or you go into PA or you do whatever you're going to do, unless it's PT and then you don't do very much of this, but you are going to need to understand that knowledge of the type of receptor precedes any drug that could possibly interact with it. Does that make sense to you conceptually? Okay. So if you go into pharmacy, you're going to do a hell of a lot of talking about receptors. So I thought I might introduce you to the 5 big categories of receptors that exist. Okay. I don't test on this really deeply, but I do want you to try to get a conceptual understanding of it. Okay. Ready? All right, so first, let's just back up and talk sort of philosophically about why things are named what they're named and what kinds of receptors you have big picture. So first, sometimes the receptor is named for like the neurotransmitter, like a GABAergic receptor, but sometimes it's named for a drug that interacts with it, like an opioid receptor or a cannabinoid receptor. And what is that about? Why is there no naming convention? It's just a little history, just a little like leftover history that's in there. So often the impact of a drug is known long before we knew anything about neurophysiology, right? So we have been using opioids for thousands of years now, and we've been using cannabinoids for hundreds and hundreds of years now. So a lot of times the impact of a drug was known long before the receptor or the chemical was known, the endogenous chemical. Does that make sense to you? Yeah? Okay, so people use, I said hundreds, but I looked it up and it's thousands actually. Okay, so the receptor though, think about this. The receptor must have predated the drug, right? Otherwise, you would have just gone, hey, I just smoked that plant and nothing happened like every other freaking plant I smoked. And then you wouldn't have smoked the damn plant again, right? So the receptor must have always predated the drug, however, meaning that despite what people tell you, the logic of, for instance, telling someone, you have cannabinoid receptors, that must mean that you're supposed to smoke cannabis. That's bass-backward logic, because what you have is you have receptors for an endogenous chemical, and cannabinoids also bind to that receptor. Does that make sense to you? Like, have you ever heard anybody use that argument? Obviously, you're supposed to take opium because you've got opium receptors. You don't have opium receptors, right? You have receptors that also bind to opium. So the receptor must have predated the drug, meaning that there are no opium receptors in the brain. There are receptors that bind to an endogenous chemical and also bind to opium. So the drug interacts with a receptor system that's already there. And if you found the drug first, a lot of times the receptors were named for the drug, so like opioid receptors. But if you found the chemical first, before you knew what interacted with it, you would call them GABAergic receptors, right? So why are they not called alcohol receptors? Because we didn't know that alcohol interacted with those receptors when we started using it. We found GABA and we're like, oh, that's how alcohol works, right? Okay, so the big picture here is that what is happening is the drugs that we are taking in are interacting with a chemical messenger system that we already had, right? Does that make sense? So, but the names are not consistent because it's based on sort of when things were discovered. So with that in mind, what we can say is that the neurotransmitter system predated the drug. But think about this. I found this idea fascinating. I have no evidence for this. I'm just telling you like a little philosophical exploration that I did in my head. Okay, so if cannabinoids and like marijuana, for instance, interact with receptors in our brain, opioids interact with receptors in our brain, those are pretty fantastic evolutionary mechanisms to make sure that we continue to propagate opium poppies and marijuana plants, right? I mean, that's pretty fantastic, right? They interact with something in our brain. And then what do we do with opium poppies? Well, we plant the hell out of them. And we take care of them, and we give them fertilizer, and we make sure that they can make some more babies. And what do we do with marijuana? Well, the same thing. And I'm like, oh my God, they have hijacked us. They are using us for evolutionary purposes, which I find really interesting and have absolutely no evidence for, but I think it makes sense. I like it. Okay. All right. So the next thing that we're going to do is signal transduction pathways. What we are going to be talking about here is the in-between part that we haven't filled in. So what we said is, okay, so there's neurotransmitters and there's cell activity, right? The neurotransmitter calls it target cell activity. What we didn't fill in is how, right? What's the middle part, right? You bind to a receptor and then the cell does something, okay? Well, what we figured out, of course, is that what the target cell does is more closely related to the receptor than it is the neurotransmitter. So for instance, if these two are receptors and I am dopamine, if I bind to a dopamine receptor that is also a potassium channel, then I inhibit the activity of that cell. But if I am the same neurotransmitter but bind to the receptor that's also a sodium channel, then I increase the activity of that cell. So what happens on the target cell is really starts with the receptor, okay? And what we call what happens on the target cell is this term called a signal transduction pathway, which doesn't mean a whole hell of a lot to anybody. What it is a game of telephone, okay? So stick with me. We are going to play telephone to try to get something to happen in the very back. Nikki's in the very back, and what we are going to try to do is we're going to try to make something happen to her. She's our nucleus, okay? I am a chemical messenger, and Mike is going to be the receptor, okay? And what's going to happen is if I am a water-soluble chemical messenger, I don't go in, right? I stay outside the cell. What I have to do is create a game of telephone that ultimately gets the nucleus, for instance, to do something, right? But he also has to stay put, right? Does that make sense? So he's a cell membrane receptor, and I'm a water-soluble chemical messenger. So I bind to him, and what he has to do is, for instance, tell Anna something, right? And tell you something until ultimately we get a protein synthesis or a secretion or something. So this is a game of intracellular telephone, right? And with a water-soluble chemical messenger, it's a long game of telephone. But if I were lipid-soluble, do I have to deal with any of you people? No, I can go straight back to the nucleus myself and force protein synthesis, right? So what we call these is we call them signal transduction pathways or signal transduction mechanisms. And they are games of telephone to get the target cell to do what you want it to do, right? It's going to be a shorter game of telephone with a blank soluble chemical messenger. Shorter game of telephone with a blank soluble chemical messenger. Lipid soluble. Longer game of telephone with a water soluble chemical messenger. So let's do the easy one first, okay? So the signal transduction pathways are responses to a neurotransmitter or any chemical messenger. Could be a hormone, could be a paracrine agent. So the first one is the signal transduction pathway initiated by an intracellular receptor. These are called gene activation. So let me introduce you to it, and then I've got an animation for you, okay? All right, so I have a lipid-soluble chemical messenger. It's going to diffuse straight through the cell membrane and find a receptor either in the cytoplasm or you can even have them in the nucleus. And what it's going to do is this hormone receptor, this chemical messenger receptor complex is going to be a different shape than either the chemical messenger or the receptor alone, right? New shape, you guys with me? That new shape will now go and bind to a region, a specific gene, right, and activate transcription of an mRNA, which will then do translation into a peptide. And now I have a new protein. And why do I care? Well, because new proteins are a new structure, potentially, or a new function. Yes? Okay, so let me give you an example, and then I'll show you an animation. So this might be, for instance, how testosterone causes you to make actin and myosin in muscle cells. Yes? Testosterone is lipid soluble, yes? Testosterone diffuses straight through the cell membrane, yeah? Binds to its receptor, goes to the gene for, let's say, myosin, causes transcription, and then translation of myosin, and then you put it in as part of a sarcomere. Does that make sense? Or part of a myofibril, right? And testosterone is better at doing this than estrogen. Right, it's better at this whole process of gene activation. Okay, so let me show you an animation that will reinforce that, which is this one. This one is also using a hormone. Not surprisingly, there are actually more lipid-soluble hormones than there are neurotransmitters, and we don't actually know how that one works yet inside the cells. Okay, so this is aldosterone, and that fits the criteria, right? Aldosterone diffuses straight through the cell membrane, binds to its receptor, and the hormone receptor complex moves through the nuclear pores, binds to a specific region of DNA, activates transcription. You guys remember all this from 1610, transcription, translation? And Mike's like, shut up, Melanie, why are you talking about this? And then translation on the ribosome. And then I have a new protein. What does that new protein do? I don't know, something different than the cell did before, right? It's either a new structural protein that changes something about the conformation of the cell, or maybe it's an enzyme that wasn't present before, right? But either way, it changes something about the structure or function of the cell. Otherwise, you wouldn't have bothered because this is energetically and also resource expensive, right? Okay, so that is called gene activation. So, and it is the mechanism that you are going to learn for lipid-soluble chemical messengers. It's not the only one, but it's definitely the most common one for lipid-soluble chemical messengers. Examples, steroids such as cortisol, estrogen, testosterone, those do this. Okay? All right, so now this is where it gets more complicated and also slightly boring, so pinch yourself or something. There's nothing I can do to make this entertaining. Everybody hates it. I hate teaching it, but it needs to be done. Okay, so signal transduction, so stick with me. Now I'm going to talk about signal transduction pathways initiated by plasma membrane receptors, meaning that I'm a water-soluble chemical messenger, and there are 4, okay? Two of them you've already learned about, just not in detail, okay? So I'm a water-soluble chemical messenger, so I have to stay outside the cell. This is going to be a longer or shorter game of telephone. Longer game of telephone. So the first one you have already met. You met it multiple times under multiple different contexts. And it's this one. A cell membrane receptor that is also an ion channel. Yes, these are called ionotropic receptors. We've met them before. Do they cause a fast or slow response in the postsynaptic cell? Fast response. FYI, just so you know, It doesn't have to just be about depolarization and hyperpolarization. Sometimes you just needed calcium inside because you needed calcium to activate something. But sometimes it depends on whether it's an electrically active cell. Like other cells don't care that much about depolarization and hyperpolarization, right? Sometimes you just let calcium in because you need some calcium. Sometimes you let chloride out because you need to get rid of some chloride. But regardless, what it's going to do is it's going to change something about the cell. So here's another one, FYI. This is the same kind of thing, but FYI, you don't always have to be depolarizing and hyperpolarizing, or that's not always the point. So a chemical messenger binds to, for instance, a calcium channel. You could depolarize or hyperpolarize, but you could also use extracellular calcium to initiate muscle contraction or to initiate secretion or to like activate some cascade inside the cell. But the important thing to understand is that you might need to do this because I can't go inside the cell, right? Because I'm water soluble. So if Mike is that calcium channel right there, what happens is I am the first messenger, but I have to stay outside. So what he does is lets in a second messenger. Have you ever heard of second messenger systems? Yeah? Very often that second messenger is calcium. It's really common. So what are you going to do after that? Well, I don't know. The calcium is going to cause something to happen in the cell that didn't happen before, right? Something. Okay? So these are ligand-gated ion channels or ionotropic receptors. Let me tell you that a couple that you're going to run into in your career will be this category. Nicotinic cholinergic receptors respond to acetylcholine and nicotine, and they fit into this category. Not surprisingly, you already met them on skeletal muscle, didn't you? Skeletal muscle has nicotinic cholinergic ion channel receptors. And in the central nervous system, these nicotinic cholinergic receptors are the ones that are involved in learning, attention, and memory, and the ones that degenerate in Alzheimer's disease. They're also located in the PNS and somatic system. Okay, so next one. Still dealing with water soluble. Now we are going to move to not an ion channel. Okay, so this is a receptor on one side, yes, and an enzyme on the other side. It's not letting anything in or out of the cell. Here's what it's doing. Chemical messenger binds to the receptor. The other side here is an enzyme called a tyrosine kinase. The reason it's called a tyrosine kinase, by the way, is look what it does. It takes a protein with a tyrosine amino acid on it and it slaps a phosphate on it. And now this is active and it wasn't before, okay? What does it do? I don't know, changes something about the cell. So now I've got an active enzyme that was inactive before. So these are called tyrosine kinase receptors. And listen again, the receptor is also an enzyme. Yes, the receptor is also an enzyme. And you run into these, they're relatively common. Tyrosine kinase receptors are really common hormone receptors for water-soluble hormones. Okay. Next one. Your textbook doesn't have a picture of this one. I don't know why, so I had to snatch one from another textbook. Okay, so the receptors that, the next ones are really, really similar to this, only this end, the receptor end, and this end are actually two separate proteins instead of all one protein. Okay, so all I can find is a bad picture from the other textbook that we used to use. So look at this one. Here's the receptor, yeah, but this is a separate protein called a JAK kinase. So these are called JAK kinase receptors. And these are so damn common that JAK actually stands for just another kinase. It does. Because they used to get so excited when they found them that they would name them Sheila's amazing kinase, give me a Nobel Prize. And then they were like, you just found hay in a barn, so whoop-de-doo. So they just named it JAK 264, JAK 782. There's so many. That does stand for just another kinase. So the just another kinase, it's bound to the receptor. What does it do? It's a kinase too, so it behaves a lot like the tyrosine kinase, and it phosphorylates a protein and then activates something that wasn't active before. But again, the difference between a JAK kinase and a tyrosine kinase is that with the tyrosine kinase, these are all one protein. And with the JAK kinase, they're two separate proteins stuck together. Okay? Now, why the hell do you care about JAK kinases? Because growth hormone binds to a JAK kinase receptor, and growth hormone tends to be pretty damn important. Okay? All right. And then the last one you've seen before, and then we're done, and then I will revisit and organize all of these in your head. So these are called your Gprotein coupled receptors, and we saw these before because these are like the most inefficient way to accomplish something ever. What the Gprotein receptors do is, these are the ones that I basically said, these are like going around your ass to get to your elbow in Texas. That's what we would say. In Utah, they don't say that stuff, and they would prefer that I didn't, but, you know, I'm here. Okay, so what happens with these is that you are going to try to accomplish something similar. You are either going to activate an enzyme or open an ion channel, but instead of just doing it yourself, you're going to use 5 friends together to do it, right? So these are called Gprotein coupled receptors. By the way, the reason they're called Gprotein coupled receptors is that they use as their energy source not ATP but GTP, guanosine triphosphate. That's why they're called that. So here's what happens, guys. My goal in this thing, in this picture, is to open this ion channel right here. Yeah? But I'm not going to do it directly. What I'm going to do is I am going to use a bunch of my friends to help me get this ion channel open. Okay? So this is a water-soluble chemical messenger. Yes? And it is going to go and bind to a receptor. The receptor is not going to do anything directly. It's going to involve its friends alpha, beta, and Mike Warren told me that was epsilon. Is that epsilon? I don't know those letters. Okay, so what's going to happen is these are going to slide through the cell membrane, and then the alpha subunit is going to slide off and smash into the other protein. If that was an ion channel, that's how you open it. If that was an enzyme, that's how you activate it, right? Okay, and then you have to put all of it back before you can use it again. Doesn't make a damn bit of evolutionary sense to me, but it is what it is, so we will have to accept it. So now I use GTP to get it all back into position, and I can't use this again until all of these go all the way back into position. And now I can use it again, right? I have no idea. I don't know how they figured it out, but guess what? These are so common. This is like among the most common types of receptors that exist. So you can't pretend they exist and stick your fingers in your ears like you all want to do. Because let me show you where you run into these. These are muscarinic cholinergic receptors, the other category of receptors, and they're active in the CNS as well. They are both alpha and beta adrenergic receptors, which you have to deal with when you're dealing with cardiovascular issues. Okay? And they are also serotonin receptors, which ends up being pretty damn important to not going crazy. And these are all this type of receptor, which is a Gprotein coupled receptor. Okay, so let's round this out and say, what happens at the end of all of this? Well, the cell behaves differently in some way, shape, or form. What could it do? You could activate transcription and the synthesis of new proteins. You could make something new. You could activate or deactivate an enzyme that was already present. You could induce secretion. You could induce cell division. You could depolarize or hyperpolarize the cell. You could change the metabolic rate. But you wouldn't have done all of this if you weren't going to do something with the cell. So let's review. Ready? Okay. There are 5 signal transduction pathways that you need to know. Okay? I don't test on them in a ton of detail, but you definitely have to have them organized in your head. The one signal transduction mechanism that you learned for lipid-soluble chemical messengers is called what? Starts with a J. Gene, definitely gene something. Somebody help her. Gene activation. Gene activation. Okay, that's the one that you learned for lipid-soluble chemical messengers. And then you learned 4 for water-soluble chemical messengers, because there are more water-soluble chemical messengers. Sorry. Okay. One of them is, for instance, the type of receptor you would find on skeletal muscle. The receptor is also a what? Ion channel. Those are called ionotropic receptors, and they're pretty fast. Good? Okay. One, the receptor is also an enzyme, the same protein as a receptor and an enzyme. What are those called? Tyrosine kinase. Exactly. One, the receptor is stuck to an enzyme. And those are what? JAK kinase, like nobody's impressed by them. And then one is the most inefficient way that I can possibly imagine to accomplish something, but apparently relatively common. What are those? Gprotein receptors. Maybe somebody at some point will teach you why they make perfect sense evolutionarily, and then you can email me and tell me and make me smart. Okay? So, Melanie, I got it. I understand why Gprotein receptors are so common, because I sure as hell don't. Okay, so the last little tidbit in this set of notes is easy and kind of interesting, and then we will move on to the next one briefly. Okay, so here's the deal. Your body has the capacity to change its responses to constant sensations. Okay, so I want you to think about this for just a second, and then we'll sort of build the story. If you work in someplace stinky, like if you work in a retirement home, you know it's stinky. Let's just all acknowledge it. Or if you're one of those poor people who decides out of their own volition to volunteer as a lab aide in the cadaver lab, right? It does not smell good in there. So the question is, when you walk in, of course you smell, and it smells bad. But after about half an hour, do you smell it at all? No, you don't smell it at all. And here's why. Your body has the capacity to change its responses to constant stimuli to save ATP. And so let's go through basically how transmission works and then talk about this concept, which is called adaptation or accommodation. So here's what happens. So you guys know that every single action potential begins with a local or graded potential, yes? Okay. And the local or graded potential could be chemically induced, or it could be light induced, right? Or pressure induced or temperature induced, although those are hard to picture. They obviously exist, okay? And then the depolarizing graded potential, if it's big enough to hit threshold, then you get an action potential. And we know that the intensity of an experience or the intensity of a sensory stimulus is not conveyed by the intensity of a single action potential, right? It's conveyed by what? The frequency of action potentials over time. Got it. Okay, so we all know that. So because in a single neuron, an action potential is the same as every other action potential, and it's the all or none phenomenon. So the magnitude of the initial stimulus is conveyed to the central nervous system by frequency, not intensity of action potential. So here's what happens. When you encounter relatively constant stimuli, then you have the capacity to turn down the frequency and to make the receptors slightly less sensitive to those. Because if it's a constant stimulus, it's probably not likely to fit into the three categories that are always worth action potentials. The three categories that are always worth action potentials are something that could kill you, yeah, something that you could eat, something that you could have sex with. Those, even if you don't want to, will sometimes send relatively frequent action potentials. You're like, my wife is right here, I'm not looking, I'm not looking, I'm not looking, I'm not looking. Or somebody goes by with fajitas and you're like, I'm not looking, I'm totally looking. Okay, so what happens is we have the capacity to adapt or accommodate out constant stimuli, but we also have a cerebral cortex that can decide what we attend to a certain degree. So here's what happens. I want you to think historically about somebody walking across the African savanna thousands of years ago, right? Proto-humans or early humans walking across the African savanna. There's all kinds of sensory stimuli coming in. You've got the sound of the water going down the stream. You've got the sounds of the birds tweeting. You've got the wind going through the rushes. And all of that information is really going in. But it's relatively constant, so it's going in relatively low frequency. Your receptors become less sensitive when it's a constant stimulus, but then very, very soft, the person hears, right? It doesn't have to be loud, it just has to be different than the stimuli. And whenever a stimulus is different, it gets frequent action potentials until it's categorized, right? Or constant. So if you think about that, that means that what's going on right now is all of the constant stimuli that you're experiencing. You have the ability to reduce action potentials based on them. So think about, you can feel your clothes on your body right now, but you're not attending to it, and you're not sending very frequent action potentials about it. But you can, with your cerebral cortex, choose to attend to it, and then you'll feel it, right? Okay, right? And then sometimes it's a little difficult to choose not to attend to it again, right? So your cerebral cortex has a certain degree of control over that. But what happens is you are set up to notice changes. And those of us who are parents definitely experience this very regularly. So how many of you guys are parents? Okay, we got a few parents in here. I want you to imagine you sitting in trying to study or get something accomplished in one room, and your kids are in the other room, and they're making all kinds of kid noises, right? There's like arguing going on, probably a video game, some noisy ass toy that your mother-in-law gave them, whatever. And then something either crashes and it's really loud, right? And then you increase action potentials, but it doesn't have to be loud. Think about what would happen if your kids were playing in the other room and all of a sudden you heard silence. All it has to be is different. So when the stimulus changes, you increase the frequency of action potentials. We are set up to spend ATP on different stimuli. That's why people can live in stinky towns. Have you ever driven through a paper mill town? It smells like somebody took a foot and wiped their butt with it. That's what it smells like. It smells absolutely atrocious. And you wonder, how the hell people live there? Well, it's the same that working in the cadaver lab. After a little while, you are not attending to it any longer, and it doesn't really, it's not like you're killing off the nasal epithelium, because then you wouldn't smell it the next time, right? What you are doing is decreasing the frequency, right? But can you still smell a fart? Yeah, you totally can, so don't try it in the cadaver lab, right? Or perfume. You can totally still smell it because those are set up. Now, the only other thing that's interesting about this is that males and females tend to, and this is not a sentence, it's a predisposition, and there's bell curves on both sides. Males and females, we tended to have evolved to have different capacities for adaptation and accommodation based on predispositions of behaviors, historically speaking, for thousands of years. So now, in this day and age, our activities are kind of overlapping quite a bit. Like, I could probably do most of the same jobs that Mike could do, and Mike could do most of the same jobs that I could do. But historically speaking, that's not the case. We didn't do most of the same things. Historically speaking, mostly humans have been relatively tribal, right? Living in groups, right? Okay? But mostly women were more likely to, in most situations, stay back with the children, and you had more kids than you intended to have because you didn't know how not to have kids, right? One of them was always trying to die of something, fall off, get eaten by a saber-toothed tiger, whatever the hell. And then you also had to deal with all of the different input from all of the other people there, right? So women tend to be predisposed to multiple foci. So you can have multiple things going on at one time. It doesn't necessarily mean you're great at it. It means that I could talk to Mike and also eavesdrop on your conversation. It's just something I could do, right? Males, however, tend to have a predisposition to single focus activities and adapting or accommodating out all extraneous information. And if you are not careful, you are extraneous information, because what they are predisposed to do is to go out and track the wildebeest, find the wildebeest, kill the wildebeest, bring the wildebeest back home, feed their family, have sex, go to sleep, do the same thing the next day, right? So what happens is they are really, really good at single focus and not super good at multiple focus. Now, this is not per individual, this is per group. And so what happens is, unless you are the wildebeest, then a lot of times you don't actually get the attention, right? And so with women, a lot of times they have trouble actually focusing on one thing when there's a whole bunch of other things going on. Now, individuals, of course, vary, but we do have those predispositions, and it's only been 100 years or so when we've been really changing our patterns of behavior. So it's still our predisposition. You kind of have to understand that because you're going to live with another person, right? And a lot of us are going to live with a person of the opposite sex. Not all of us, but a lot of us are going to live with a person of the opposite sex. They are not like you. They are kind of the same species, but with an asterisk, right? A little bit different. Okay, so here's what I want to tell you about what we're going to do next, because you are going to have something that I want you to do over the weekend. It's not major. What you guys are going to do next is we are going to compare the somatic motor nervous system to the autonomic nervous system. But what I'm going to do over the weekend, I'll probably get it up tomorrow, is I am going to give you a 30-minute prep and review video with a little worksheet for you to fill out, because what I need you guys to do is to get comfortable with the terminology that you've already learned and people suck at, which is you need to know what I mean when I say autonomic, efferent, afferent, sympathetic, parasympathetic, right? And in my experience, that vocabulary sometimes inhibits people from understanding the physiology. So I'm going to do the first part of this notes, just organizing it, and a little worksheet, and then you're going to come in and then we're going to do the real phys. Good? Okay. What'd you say? No quiz. It's just going to be like a worksheet. I'm not going to take the worksheet either.